Oh, I think we have to proceed with yours, and then we could have Pedro continue his talk after your talk.
Okay.
So, is
Yep.
that okay? And
Yep.
I could introduce you at this point. Okay. So, sorry we lost an entertaining talk there from Pedro, and we learned a lot about blood pressure somehow, and not eating too much salt. That's what my doctor is saying. But we'll go to Carlito's talk, and let me introduce Dr. Lebrilla. So some highlights from Dr. Lebrilla's very long record. He got his BS in chemistry as well as his from Irvine, UC Irvine, and then his PhD from the University of California at Berkeley. So I noticed that Carlito is a real Californian. After his PhD, he got his postdoctoral fellowship, the von Humboldt postdoc, the NATO-NSF, and then the University of California President Postdoctoral Fellowship. These are very prestigious postdoctoral fellowships, and very proud of you. He also got an NSF extension for creativity. I think that was just before he became an assistant professor. He has been a member of editorial boards, and he's also a co-editor of one of the journals in his field. He is an elected fellow of the American Association for the Advancement of Science. This is a very prestigious honor, and not very many people gets this award in the US. He was the 2011 co-award in science. So he's the third one speaking today, and he's the current chair of the PAASE Board of Directors. I look at Dr. Carlito's vita, and there's a very interesting highlight there. So he went, of course, as an assistant professor at the University of California at Davis. And then after a few years, he became an associate professor. And I think within less than a year, he was promoted to full professor. And I could tell you that that is a fit that doesn't usually happen. So I put here that he has one of the speediest promotion from associate professor to full professor. He's a distinguished professor in chemistry at UCD, and he's also a distinguished professor in the biochemistry molecular medicine, again at UCD. He has been the chair of the Department of Chemistry at UCD. He has been the executive board of the American Society for Mass Spectrometry, Academic Senate Faculty Distinguished Award at UCD, and he has been a chartered member of an NIH study section. So that's usually a 3-year appointment, and you have to sit on every meeting of this study section. He is also one of the founders of a company that is the topic technically of what he'll be presenting today. And this is Evolved Biosystems, and I think it's still ongoing. One of the highlights of Carlito's records that he has so many publications, at least 320 publications with at least 25,000 citations in an H index of 86. So that's very impressive. And we are very proud that he is with us, and he will be giving a talk on carbohydrates in the next generation of bioactive foods. So Carlito, it's your time to give your presentation, and let me unshare here.
Great. Thank you very much, Ed. So again, it's always a pleasure to be a part of PAASE, and particularly in this time when we're all sort of sequestered, it's great to connect in this way. Let's see. All right. So I'm hoping everyone is hearing me and that you can see my slides. I'm going to talk about something that's sort of different and something that we've been heading towards without realizing it, but and that is the future of food. And if you think about where food is and where food was, if we call food 1.0 during the hunter and gathering, the hunter and gatherer phase, where we essentially found food, we looked for it, and we weren't very picky. It's food that we could find, food that we could kill, and food that we could eat. Food 2.0 is the greatest thing that's ever happened to us in the sense of being sufficient, self-sufficient. We've had, we could grow our own food, we could grow as much food as we want. In fact, oftentimes we waste too much, but there's just so much food. And all of this is really just accessible because of agriculture and how far we've advanced it. And we're now actually in the beginning of a new phase, and that's what we call, we could call that food 3.0. And this is a much more precise way of nutrition, and much more personalized. And so the next invention in food is really going to be towards personalizing and trying to figure out which food is actually the ones that are best for us. And a lot of this is coming about because of what's the so-called omics revolution. And the omics revolution is really changing health, and it's changing how we see food. And when I mean omics, I'm talking about the genomics, of course, but really proteomics, transcriptomics, metabolomics, lipidomics, and so on. And a lot of these omics takes a lot of really specialized and very expensive tools. And so these are the instruments, the coterie of instruments that we use in my lab for doing advanced omics. So we have an Orbitrap, we have a QTOF, we have, actually these are two different instruments, but I forgot to change the picture. So we have a NanoLC with a time of flight, NanoLC with a QTOF, and then two triple quads, and soon we're gonna get another instrument. And when I was in the Philippines last time, someone said, someone asked me, why do you need more than one mass spectrometer? And I was advocating that we should have more mass spectrometers in the Philippines. And it's because they do many different things. You can't have just one because you need all of them. You need a lot of different ones just so that you can do different compounds. And so I would still advocate that we still need more mass spectrometers in the Philippines. But it's expensive. So we name our instrument. So the Orbitrap, we call the Orbitrap. And that's a $1.2 million instrument. The QTOF is 650,000. The ChipTOF is 500. So we name them after TV characters like Jon Snow. This one we call Jon Snow because he keeps coming back from the dead. And then we have our triple quads. And the point of this is not that there's a lot of money there, but the point is that research is expensive. And it has to be supported in that way. And each of these instruments require their own infrastructure. And so to really be able to do this kind of analysis and these types of omics, we really need the infrastructure that allows us to do these things. And so I'm going to start this by saying, if there's a food that we should be eating, it's human milk. So right now in the table, there's a war going on. Some people think we should eat only meat. Some people think we should only eat vegetables. Some people think we should be a mixture of meat and vegetables. And so there's this war about what we should eat because we don't actually know what we should be eating. And so we studied milk for many years now, and I've given parts of this lecture before. But it's an important concept because milk really is the model for the perfect food. And so if you look at what's perfect about milk, what is it? What's in the perfect food? Well, you have lots of carbohydrates, you have lots of fats, you have fiber, you have proteins, and you even have peptides, and then you have minerals and salts and so on. And so a food that has evolved with us is made up of really basic components that we actually already all know, right? Now, it used to be that we think that food was mainly for nutrition, it's for growing cells. But we know that's not really true anymore. That's such a limited understanding of food. We know, for example, that food can actually train the immune system in infants. We know the right food, for example, we saw dance lecture on vaccine and COVID. And the senescence is a key word there, because as you age, your immune system doesn't react as well as it does when you're young. And so understanding how the immune system is built up when you're young, particularly through nutrition, is really important. One function of the food is that it feeds a gut bacteria, and not just one bacteria, but it feeds sort of our constellation of many different types of bacteria that without, we would certainly, if not die, we would be all quite miserable. And brain development. So there are some great experiments that show that having the right components of milk can actually develop the brain. And so from nutrition, which is really a very small component of what food does, to a greater understanding is really what we want to do. This is really where we want to be. We want to know what's in food, how it helps us. But primarily what we need to know is what are the structures of food. And so if we go back to human milk, one of the things that really made all this possible was our ability several years ago to characterize every single structure of this fiber that's found in human milk. It turns out human milk has these fibers that are called human milk oligosaccharides, and for the longest time, we didn't know what they did. And the reason why we didn't know what they did was we couldn't tell what the structures were. And so if you look at their structures, it's presented this way in the liquid chromatogram, mass spec chromatogram. And if you look at it as individual structures with the size corresponding to their abundance, it looks like this. And it turns out there's actually two types of human milk. One group of mother will produce these kinds of human milk, and the others will produce this kind. And what we found was that if you put this mixture into this one very specific bacteria that we discovered, all these abundances will disappear. If you put them in a different type of bacteria, it turns out they don't disappear, and they just go all the way down and become feces, right? And so we discovered that there were about 300 of these structures, and we've characterized them, and we published several papers that really just characterize what these structures were, how they interact, and how they actually are beneficial to this one bacteria. The other thing that we found was that depending on where the mother was, or depending on where you grew up, where your ancestors were from, there's a certain fraction of people who have either this kind of milk or this kind of milk. So this kind of milk up here is one that's called a secretor, and the one down here is called a non-secretor. And that's represented here by the secretors as being blue and the non-secretors as being red. And so it used to be that people would say there are 20% non-secretors, but that's because that's what the European population was. And so because a lot of the research was done by Europe and the US, everyone thought that there was only a 20% secretors. But when we studied milk from all over the world, involving thousands of samples under the Gates Foundation, what we found was that the ratio of secretors to non-secretors differed depending on where you were from. So for example, in the Philippines, there are about almost 30% non-secretors. If you go to Poland, it's about 20%. However, if you go to Africa, in South Africa, it's almost 40%. And India has really high levels of non-secretors. On the other hand, if you go to South America, say in Peru and Bolivia, you find that there are almost no non-secretors, and they essentially have been pretty much wiped out from the population. What we also found was that this important bacteria that feeds on human milk was disappearing in the United States. And you could see here in Davis, California, only 25% of the infants had them. In Ireland, about 20%. But in countries like Bangladesh and the Gambia, you see that they're almost in 80% of the kids. And so what we did then was we started a company to bring back essentially this bacteria so that people in Davis will have, and in general, those who are deficient in this bacteria will have them. And that's the company that we started. And I think on Wednesday, I'll talk to you about my experiences with starting companies. But anyway, the company itself is now on its way to a Series D a Series D. You can buy this in different hospitals. So far it's raised almost $100 million in venture capital. And Science wrote an article about this a couple of years ago, and they called it Nature's First Functional Food, right? And so this is my collaborators who are co-founders of the company. David Mills is a microbiologist, Bruce German is a food scientist, and we're the analytical chemists who sort of identified what these structures were. And so this is the product. You can actually buy it on the web. It's now also available in hospitals. This is they're performing clinical trials on infants and in the NICU, those who are premature infants. And we think that if you go on their website, you see the mothers really rave about what this product can do. So the other thing so there are other components of human milk, and the other ones are the proteins. So there are a lot of proteins in human milk, and one of the more important proteins, well, the more important protein is called lactoferrin. And we studied this protein also, and what we found out was that this protein is modified, it's post-translationally modified with this glycan. And when you put this protein in the presence of a bacteria like Salmonella and E. coli, what we found was that this protein blocked the binding of different Salmonella. So this protein was actually protecting the infant from getting pathogens. And when we started clipping off these different glycans, different structures from the lactoferrin, its ability to block also differed. And so it turned out that this is actually what's causing the lactoferrin to block the binding of Salmonella. And if you make if you cut this off, then it no longer blocks binding. But the other thing that we found was that during lactation, this protein was actually changing its glycosylation. And so it was sort of changing to address the changing variations or the changing populations of different gut bacteria. And so this was actually then a very targeted method whereby the mother was protecting her infant from different bacteria that is altering during the course of lactation. The other thing that we found is that human milk was also full of peptides. And so you could take a protein like beta-casein, and each of these lines represent all the different kinds of peptides that we see from beta-casein. We also find it from all the other proteins that are there. And so no one had ever seen this before. And we wondered why there were peptides in human milk. And what we found was even more protection for the infant. And so what we did was we also developed tools, peptidomic tools, that allowed us to quantitate the site of glycosylation, as well as the proteases, the site of these peptide formation, as well as the protease that are involved in producing them. And when we started picking up some of them and throwing them at bacteria, what we found was that they killed bacteria, right? And in fact, 62 peptides had about greater than 57 sequence overlap with known bioactive peptides that are antimicrobial and those that modulate the immune system. So in this, what we found was that milk wasn't just nutrition at all. In fact, nutrition was a very minor component. Nutrition was lactose. Everything else was really to protect the infant. They protected it by killing pathogens, by blocking them from binding, and by actually feeding the good bacteria that can outcompete the bad bacteria. And so we're actually doing a little bit of COVID research, and we're now seeing if some of these components will actually then block COVID binding. Because if you look at infants who are breastfed, they don't seem to be getting the virus. So there's, again, more protection that the mother has. And so now we know, again, as we said, that food is really nutrition, but that is just such a minor component of food. And what made us realize this was that knowing that the structures, what the structures of food are. So the structures of food is important. And so now I put in front of you, so I love eggplants. And if you think about eggplants, there are just so many different varieties. And this is just a small number of them, right? There are Filipino eggplants, there are Chinese eggplants, there are eggplants from Europe. I'm growing this in my backyard, these white eggplants, and they're really good. But really, these are all the same species, but they're all very different phenotype. And so what you're seeing here is really sort of the post-genome. The genome told us a lot, but we need something more now. What we need is really what differentiates not just species, but the individual, right? So I like to think of food really as the post-genome era. We're really officially in the post-genome era, right? And so if we look at food again, remember I showed you what the component of milk was. Well, it's composed of carbohydrates, proteins, fats, and metabolites, right? And also minerals. Well, we have tools for all these. Metabolites, we do metabolomics. Fats, we do lipidomics. Proteins, we have proteomics. What we're stuck on is carbohydrates. What is the structures of carbohydrates? And despite the fact that carbohydrates is the most abundant fraction of nearly all foods, except perhaps for meat, we know, or at least for all diets, we know actually so very little about it because we don't know its structure. And so we started training in our expertise in human milk oligosaccharides to look at the carbohydrate structures in food. And it's much more difficult. So carbohydrates in food can be made up of monosaccharides. It could be made up of oligosaccharides, which are chains of monosaccharides, and it could be made up of polysaccharides. And in food, you have all these different permutations. So you can have large polysaccharides, you can have oligosaccharides, you can have monosaccharides, and all these things will behave differently and will have their own very special function in food. And so remember when I showed you the milk oligosaccharides and we found out what they did, right? That research took 80 years. So in the 30s, these milk oligosaccharides were known, and they were known to be there, but only recently did we figure out that they're actually just food for the microbes. And it was because that we were able to determine what these structures are, and the structures are very complicated. And so as humans, we always think, well, we're much more complicated because we're more superior to plants. Well, as many of you know, plants actually have more genes than human. And if you look at the carbohydrates, their carbohydrate is actually a lot more complicated than ours. So this is the lowly potato. Potatoes actually have very different types of polysaccharides with many types of monosaccharides. And as you can see, these things go on for millions and millions of units long, and it's far more complicated than breast milk. And so the question now is, how do you determine what these structures are? Well, we've solved this problem recently, and so a lot of the publications are now really recent. And the way we did this is we prepared a sample, we acid hydrolyzed them, and then we labeled them, all in a 96-well plate format, so we can do this in a rapid throughput analysis, do liquid chromatography, use a triple quad, which allows us to quantitate, and then in a 7-minute run period, we can see all the different monosaccharides. At this point, we can look at 22 monosaccharides and monitor them. And there's only about 15 that are naturally occurring. But it's in a 7-minute run, we can see all these different things. And so if you could do this rapidly, and if you could do this using this 96-well format, you could pretty much look at any type of foods. And that's what we did. So this is over 1,500 foods arranged. So each line here represents a sample, okay? And blue here is glucose. And so this is low glucose food, and this is high glucose food. And each of these different colors represent different monosaccharides. And so now we know what the monosaccharide compositions are of the food that we eat. And so if you want something that's rich in glucose, you can eat these foods. If you don't want rich in glucose, but say rich in galactose, you can go in this area here if you want something with fructose and so on. If you cut a slice of that, this is what it would look like here. Okay, so and some of these things are pretty exotic. So California, of course, is very diverse, and so there's a lot of different ethnic markets here. And so some of these I don't even know. My students picked them out. But for example, here's let's see, here's small red beans. Here's some I don't know what that is, sorry, bell peppers, ginger. So we also sometimes take food apart and we'll look at the peel separately and we'll look at the flesh separately and so on. But in any case, you can do that. And when you do this, you can actually use some organizing statistics, and you can group food differently. And so this is how groups fall into different patterns. And so these are foods that are high in glucose. And so that includes, for example, tapioca, rice, and also a lot of certain milks and so on. On the other hand, if you look at this group here, then it's made up of the butterscotch seeds, for example, fig skin, and so on. But what this allows you to do then is that you can sort of tailor make your food depending on the groups that you want, right? So for example, oh, here are yam leaves here, yellow cherry pits. So some of these are pretty funny actually. But in any case, so now we can separate food into different diverse groups. And so we can then answer sort of a lingering question on food. So as I said before, we disagree a lot on what we should eat, but we all agree we should eat more fiber, right? And so there's a USDA guideline about eating more fiber. The problem is, what's fiber? If we don't know what the structure of food is, how can we tell people to eat more fiber when we don't actually know what fiber is, right? And so, for example, if you look at a Yukon Gold, this is my favorite potato. You can see that it's made up of glucose, but if you look here, you have galactose and you have fructose and a lot of other things. Now, on the other hand, a red potato has a different composition, and so is a sweet potato and so on, right? So if I were to get fiber from any of these, even just one species, the different potatoes, the different cultivars of potatoes, I'm gonna get different fiber, right? So the question then is, what is fiber? And now we can answer that. Well, it depends on where you get it from. If you get fiber from a Yukon Gold, it's going to be a different composition. And should that be the fiber that you're eating? Well, we don't know. But now we can do experiments to figure that out because we know the structures. And there's a lot of fibers that are actually sold. You can go to any grocery store here in California, and you can, and also health stores, and you can see all these different fibers. If you look at the monosaccharide compositions of these fibers, what you find is that they're actually the same. And these are fibers that, because they're, doesn't mean they're good for you, it just means that they can mass produce them. And so some of these fibers are all galactomannan, some of them are sort of what's called inulin and so on. And so what we need to do is to figure out what fiber is the ones that we need and when do we need it. And then those are the ones that should be targeted for the right health, right? And so if you look at the potato, I showed you that there's the composition, but what's really important is the linkages. So if I take these two glucose units together, starch or amylose is really alpha 1,4. That means it's linked through the 1,4 carbons in an alpha position, right? If you look at cellulose, which is paper, it's beta 1,4, right? So just that change in linkage makes it a nice muffin versus a stack of paper. And so those linkages are really important. And so we need to know what those are too. We just don't need the monosaccharide composition. We need to know how it's put together. And so we developed that method too. And so now this is the same graph, except now the blue here is glucose, and different shades of blue is different linkages. So for example, the dark blue here is a terminal glucose, and then some of the different blues are 1,4, 1,3. And so we've created a library where we have 100 different linkages, and now we can do this and we can characterize food. And so now we not only have the monosaccharide composition, but we also have the linkages that make it up. And why is that important? Okay, here's different rice, right? This tells me that this is that's the fraction that's on the terminal, and this is 1,4. So this is a long starch, and this is how it's stopped, this terminal glucose. And so this kind of rice has a lower amount of branching than this, because this one has a higher level of terminal glucose, right? So that means these two rice are going to be quite different. However, it's still starch because you have this 1,4, 1,4 linkage and a lot of other less linkage. This rice here, one of my Italian colleague brought it back to me, and it's a totally black rice. However, if you look at it, it doesn't have the normal signature. It has the 1,4 and the terminal, but this is 1,3. So if you were raised on this black rice compared to this rice, or compared to this Thai rice, for example, or basmati, your microbiome will be quite different than that of those grown here, right? Or those raised with these different types of rice. So the type of rice that you have could be quite different, and you would sort of have cravings for that rice because that's the thing your bacteria is telling you to eat. And so you've heard a lot about how the microbiome is really important. You heard about how we should be, there's a lot of research now on the microbiome in the newspaper, and almost every week in Science or Nature, somebody publishes a paper on microbiome, right? So the state of the art in the genome is the tops, right? I mean, you can't, I'm convinced in the future, next to your toaster, you're going to have a sequencer so that you can see what kind of things your genes or bacteria are in your kitchen, right? Where we fail is the tools for carbohydrate analysis, right? And so we could tell you what's in your gene, but if we can't tell you what carbohydrate feeds the bacteria, or we know what genes the bacteria have, but if we can't tell you what carbohydrate structures that correspond with, all we have is the bacteria. And so that's why we're trying to characterize these things, because what we want to do is we want to partner, we want to pair the glycome, the carbohydrate structures, with the specific bacteria that is in your gut. So we want to pair the glycome with the microbiome, and to me that is where personalized nutrition is going to be when it reaches the full nutrition. Okay. All right. However, there is one final problem in this, right? In that final problem, I told you what the monosaccharides look like. I told you what the linkage look like. One thing we don't know is the carbohydrate structure. If you have a long sequence, a polymeric structure that looks like this, how can you analyze it? And the only way to analyze it is if we break it apart and we make short oligosaccharide chains that we could then analyze with our liquid chromatography mass spec method, right? However, there's no method to do that, right? Until recently. So what we did was we developed a method that we called FitDog. And this is actually just in press at the moment in Nature Communications. And what it is it's a chemical-based method using Fenton's chemistry, a very old, 100-year-old chemistry, where you take this long polymeric carbohydrate material, you give it Fenton's chemistry, and then you produce this oligosaccharide chains. And in these oligosaccharides, you can then analyze and you can determine what their structures are. And then you could put it together and give you what the polysaccharide composition or the polysaccharide chain looks like. And we call this FitDog, right? So this is the method itself, right? So you take a polysaccharide and iron and oligosaccharide. And so there's a little bit of whimsy here. We have two dogs. We call Reggie and Lucy. See, Lucy we call Bat Dog because her ears look like a bat. She has these gigantic ears. And Reggie is sort of chubby, so we call him Fat Dog. And I thought, hey, wouldn't it be cool if we named our method Fat Dog? And so we actually called it Fat Dog for a few months and even a year or so. But then, because this was a project funded by Mars, the Mars Corporation, and they wanted to use this as this method to make dog food. We said, well, they said, could you change the name? Because we don't think that would match very well with dog food. And so we said, okay, well, let's call it FitDog. So it's called Fenton Initiated Dissociation Towards Dissociation to Oligosaccharide Groups, right? So anyway, that's what fit dog means. And if you see it in the paper, that's where it came from. So anyway, so now what we're doing is we're looking at all these different sort of nodes, and we're looking at different groups of these things. And what we're starting to do is we're now feeding these things to bacteria, because in the end, what we want to know is if you're eating mainly group 4 kind of food, what is your bacteria? And if someone says they get ill, they take antibacterial, you wipe out your bacteria, how do we get it back to you? And what food do you eat to do that? So in things like Crohn's disease, irritable bowel syndrome, we think this is going to have very therapeutic value. And so that takes me then to this. There's many commercial probiotics, but how effective are they? And the problem is it's the Wild West out there in probiotics. A lot of the probiotics that are commercially available are sold because they can be easily grown. So a lot of them have very little to no clinical data. And I think once we know, as we know what the structures are of the carbohydrates, and that's what these probiotics eat, then we'll be able to pick the right mixture of what we call symbiotic, the right probiotic, and its complement food, so that we can treat people with different stomach issues. And the last part of this is sort of newer work that we're doing. And it comes to sustainability. If you're eating corn, right? This is a corn plant, and lots of corn is grown right outside of Davis. There are fields and fields of corn. If you eat corn, you're eating this, but you're eating such a small part of the plant. And usually they harvest this thing, they get out the corn, and they throw the rest of the plant away. Can we eat more of the plant? Why can't we eat more of the plant, right? Well, it's because, in my opinion, we don't understand the structures of a lot of the rest of the plant. And so we give it to cows, or we make biogas out of them. But our contention is that a lot more of the plant that we eat should be edible. And so we've started mapping out the carbohydrate composition of corn, of a whole plant. So we go all the way from the root and the various parts of it. And so the idea being, if we could characterize the composition of the carbohydrates, we can identify areas that are actually as edible as the corn. And not only that, we give a target to the breeders to figure out how to make the right kind of corn that not only gives you the ear, but is edible in different places. And so that's what this project was. And so we systematically cut up corn, and we just looked at it, at the different parts of it. And we did, because our methods of analysis are very high throughput, we can characterize all these different parts. And so this actually started off as an undergraduate project. So a lot of undergraduates do research in our lab. And so we had this one undergrad who knew this farmer. And so she went to his farm and actually picked out a number of corn. And then this then became this. So, and then this got analyzed. And so now this is actually then the composition, in this case of just the stalk here. But you could see here the different monosaccharide composition and so on. You can also pick out the different parts of the kernel. We can analyze the corn husk. You can look at the different inner versus the outer. We can look at the hair and so on. And so you can see then all the different composition. And again, the idea being, can we find other parts of the plant that we can eat? And if we can't eat it now, can we change it in such a way that it would become edible, right? And again, if you go down further into the roots and so on, and that will then be, we could analyze all that. So mapping, as I said, the carbohydrate content of plants will allow us to eat more of the plant. And so in so doing, can we do more sustainable farming? Can we create foods and plants that are where the whole plant is actually eaten and not just the part that we eat now, right? And so with that, I'd like to thank you all for being here. We receive a lot of NIH funding to look at this. We also get funding from the Bill and Melinda Gates Foundation has been quite generous in funding a lot of these sort of even crazy ideas with regard to food. And also we have a lot of corporate sponsors. And with that, I'd like to thank you all for your attention.
Thank you, Carlito, for a very informative talk and this idea of precision nutrition or personalized nutrition. Very, very interesting. Now, before